Thank you. Okay. So I fall into the group talking about novel therapeutics in the same way that Mark from Monorix was. There's a lot of work obviously going on with sugar, with metformin, but ultimately we also want to investigate whether we can invent new therapeutics for patients with some of these diseases. And that's the work that Agios does. It's, again, as Mark said, it's very early in that process to talk about our work, and we don't have much to share on CDG, but I thought what I could do is give you a sense of who we are and what our vision is, how we think about inborn errors of metabolism and treating them, give two specific examples from our pipeline that give you a sense of how we might be able to treat certain diseases like this using small molecules, and finally talk a bit about our work in CDG, and then close with some thoughts about what this community means to the fight for new therapeutics. And I think just to preview that answer, I think that this two-day process has really shown me that the CDG community has tremendous momentum and force coming together, and that is a critical piece of the puzzle in developing new therapeutics and moving us forward. So I think congratulations to everybody here, and certainly to Vanessa. This is exactly what is needed. So to start with Agios, I think the most important thing to understand Agios is what motivates us. And so we're in Cambridge, Massachusetts, and if you visited us as you walked into the building, the first thing you would see is a mural, which is shown here, which has faces of family members and friends of Agios employees who were affected by cancer. And we put this on the wall as a way to remind us every day of why we come to work and what we're trying to do, and of the fact that the most important thing that we can do with our careers is to alleviate human suffering like this. And we feel very strongly about that, and that is what drives us forward. So how do we think we can do that? So our scientific approach is to look at a field we call dysregulated metabolism. So this is looking at the cell's metabolism. So every cell has 2,000 to 3,000 metabolic enzymes. You've heard a lot about those. And those take various metabolites like mannose or mannose 1P or mannose 6P, and they change them and pass them down these pathways, all to do a couple of different things. At the end of the day, what metabolism is doing is it's taking in nutrients of various kinds, and it's turning them into one of two things, either energy or building blocks for other pieces of the cell to make another cell, or to make pieces of things that the body needs, such as glycosylation. So Agios is very interested in ways in which metabolism is different in a disease cell instead of a normal cell. Okay? And can we make a therapeutic that goes after those differences and tries to correct them? So we're focused in two areas. Our first area was actually cancer. So we study cancer metabolism, which is the ways in which cancer cells have very different metabolism networks than normal cells. And it's true, they do. And we think that by targeting the metabolic pathways that are different in cancer cells, we may be able to help cancer patients. And we think that's going to be a big wave of new cancer therapies going forward into the future. But more recently, we've gotten involved in the inborn errors of metabolism. And as you'll see later, this is partially because a couple of our programs look like they may help patients in this area, and we've gotten to know the space since then. But the more we think about it, the more we think that, of course, these are the diseases where metabolism is changed fundamentally and is driving the disease. And we think that some of our ideas may be able to help. And so we're doing as much as we can to learn about the space and to get to know all the people in it. Our vision is to transform some of these diseases and the patients who face them and their lives through scientific leadership in these two fields. So this is our summary. I won't go into it in great detail. As I've said, we're working in a new area of biology. We have an experienced leadership team. And what I'll add to that is we do a lot of partnering outside of the company. We work a lot with academic labs and with clinicians and academic sites to try to get the best science around a new program and move things forward for patients as quickly as we can. We do have a pipeline coming together, and we're reasonably well financed for a small company. This is the pipeline, and again, I won't bore you with a ton of this detail, other than to say, again, we're working in cancer as well as inborn errors of metabolism, and I'll just highlight two programs today. One is AG221, which is an inhibitor of a mutant protein called IDH2 in metabolism. And that's for cancer, but it turns out there is a rare inborn error of metabolism featuring that mutation. And so this was the first thing that pulled us out of the cancer space and into the inborn error space. And I'll tell you that story. And that'll be in the clinic very shortly. And then I'll tell you a story about AG348, which is an activator of a deficient protein called pyruvate kinase that we think will be useful for patients with a rare inborn error called pyruvate kinase deficiency. And that also is going to head into the clinic very soon. So I think those two will be good examples of the kinds of small molecule therapies that we think are possible when you want to target metabolism and change the defect that's driving a disease. Then, in both spaces, we're very active in looking for other places where we can find new targets or new diseases where these kinds of approaches may be able to help. And the CDG activity that we're doing now is very much a part of that. This is a horrible slide that no one is expected to read, but it summarizes all of the people on our management team. But I want to highlight a couple of people and make a point about the team that Agios has. One is our CEO, which is David Schenkein. He is a hematologist oncologist. He was the former head of the Tufts Cancer Center. He still sees patients once a week, and he helps anchor that patient focus, I think, at the company. He was then head of oncology at Millennium, where he developed Velcade, and then head of oncology at Genentech. And one of the principles that he's very famous for and talks a lot about is when we start to develop drugs and when we get to therapeutic development, we really want to focus the development only on the patients who are most likely to benefit from the drug, usually by selecting them based on genetic or metabolic biomarkers. That's critical, because we want to know really early if the drug is going to help people, and we want to focus that. In addition, our chief scientific officer is a chemist, Scott Biller. He was the head of chemistry at Bristol Myers Squibb and at Novartis, and he has a number of metabolic drugs that he's invented. And he and his team are key to say, you can have an insight into biology, but if you can't make a pill that has the right properties that can fix the metabolism the way we want in a convenient way for patients, then we're not going to achieve our vision. And so he's quite critical as well. We also have a growing IEM advisory group, but if anybody's interested in helping out, let me know. And the one person I'll call out and embarrass a little bit, you heard from earlier, which is Dr. Hudson Freeze. I think it was a year ago at a conference, I was at a panel talking about orphan drug development, and HUD was on it, and he was telling these crazy stories about very severe diseases that were potentially curable with sugar. And it was a fascinating story, and as you've all heard HUD speak, it was a fascinating speech. And it just riveted me, and it sounded very familiar to the kinds of things we were beginning to talk about. And HUD has been incredible in orienting us to the space, introducing us to a lot of people, and getting our research labs started in sort of being able to do some basic work in this field. And so can't thank him enough. So how do we think about the IEM space? So as you all know, this is an old field, but new in terms of therapy. So these are inherited mutations. They're severe diseases, often pediatric, with very diverse pathology. That's certainly true in CDG. Most don't have real disease-modifying therapeutics. There's diet, there's other things that we can do, but most don't have real good therapeutics yet. There's a small group that does. That's the lysosomal storage disorders. The best way to treat an IEM would be to just replace the enzyme that's defective, and that's enzyme replacement therapy. The problem is the only place we know where to do that is the lysosome through a trick in which the lysosome will actually grab the enzyme and pull it into the cell. That's great for those diseases. That doesn't work for the other 560 diseases where the mutation is elsewhere in the cell, as far as we know. And so Agios is very focused on those other diseases and the fact that there are so few therapeutic options for patients, and certainly, again, CDG is in that space. So our thought, and similar in some ways to what Mark was talking about, is can we use small molecules to treat some of these diseases? And can Agios use its deep understanding of metabolism to identify the right kind of small molecule approach that might be able to help these patients? And the main advantage, of course, of a small molecule is that it can go into the cell wherever the mutation would be, and that it can cross into the brain and help whatever extent of the neurological manifestation we can help stabilize. So a little bit about how we do this. We've built a bunch of different groups of people over the last 5 years: biochemistry, biology and genomics, metabolism, all trying to find the right places to go. Then we have a very good group that can build drugs against metabolic enzymes. And finally, as I said, we focus a lot on genetically defined patient populations and identifying the biomarkers we can use to tell us, is the drug going to work early in the clinic? We do a lot there. But let me zoom in a little bit on the metabolism story and talk a little bit about how we think about metabolic diseases and studying them. So we've built a certain technology called metabolomics, which is basically a bunch of machines that can take a lot of different inputs from a cell or from a mouse or from a patient. And what we want to say is, okay, remember the cell had all these metabolites in it. We want to break the cell open and say how many of every kind of metabolite is in the cell. And what can we learn about metabolism by looking at that? And so we can take one of two pictures of that cell. So the first is what I would call the snapshot view, okay, which is a static image of how many metabolites are in the cell at any one time. And my analogy that I'll use is, imagine a parking lot, and the snapshot view is like counting all of the cars in the parking lot and how many of each kind of car is there. Very useful. And in some IEMs, that tells us a lot of information and puts us a long way towards understanding the disease. But we also know that metabolism is very dynamic. It changes very fast, and things channel a long way through the system before they come out the other side. And so sometimes you need to take a more deep look at the system. And so what we can do is we can do a time-based view of metabolism. And this is following metabolites through the pathways and understanding the flow and the flux through those pathways to better understand the disease. And in the car analogy, this would be like saying, I want to know how many cars are moving along a highway over a given day, okay? Or how fast is the traffic moving? And I care a lot about whether the traffic pattern looks like this or looks like this, okay? There's a lot of cars on the highway in both instances, but I really want the first picture, not the second picture in most cases. And how do we do this? What we can do is we can take nutrients like mannose or glucose or other things, and we can label them using radioactive isotopes, feed them into the cells, and then take a series of time points and track it as it goes through. And this technique has already been made famous in this field by labeling mannose, for instance, and tracing it down the mannose pathway and identifying where the flow is breaking down and not passing through, and that's critical. So then how do we think about getting to a disease and a therapeutic? So this is an illustrative pathway. You start with metabolite A, and you're going to go to metabolite B and then metabolite C, and a couple of enzymes in metabolism that are helping it along. And if you have a deficiency in enzyme 2, okay, then you often get a disease from one of two things. Often it's either the buildup of the precursor, metabolite B, or the deficit of downstream metabolites, either C or some downstream product of C. And so what do we think about doing? Well, and so obviously back to my analogy, this is a traffic jam up here, which isn't good, and we aren't getting any traffic down here in the highway. So what can we do about that? So there's a variety of different ideas emerging using small molecules, pills, that may be able to help in this situation that we're thinking a lot about. So one is, if it's just about the toxicity of metabolite B, what you can do is just try to stop the body from making it. Okay, so you block its production. And so you can do that by inhibiting, in this case, enzyme 1. All right, that's sometimes called substrate reduction therapy, and in other types of IEMs, that's being explored. And I'll tell you a story about one of those in a moment. Slightly more interesting is if you have some residual enzyme 2 left with some amount of activity, can you actually bind it and reactivate it or boost its activity and get more flow through the pathway? And that's very similar to some of the ideas that Mark mentioned around PMM2 and that have been described in the literature. An alternative is if you really have a good insight into the whole network of the metabolism, can you find other places to intervene to reroute or maybe make a detour in metabolism to pass or at least alleviate some of the pressure in the system? And I think a fourth that I don't have here but has been mentioned is, can you go past the defect and actually just supply metabolite C? And the MAN1P idea is an example of that. So I think the main point I wanted to make with this slide is there's a bunch of ideas that actually are out there, and I think we want to systematically go through these ideas and understand which of these approaches can potentially be useful in CDG, as well as in other diseases, and narrow it down to the best ones and then move really quickly against those. And ultimately, the point we're trying to get to is, as much as possible, a nice healthy traffic pattern through the pathway that as closely as possible gets back to what a normal cell should look like. So now let me tell you two stories that aren't CDG stories, but that give examples of this approach and examples of how therapeutics in these diseases might come about. So one is, as I said, the cancer story. So there are mutations in metabolic enzymes called IDH1 and IDH2 in cancer. These were well known. And it was Agios' group, using the technologies that I mentioned, that actually figured out what was going on with those mutations. So the normal mutation turns a metabolite called isocitrate into the metabolite alpha-ketoglutarate. But when you get the mutation, it turns out you only have one copy of the mutation. It turns out what that does is it picks up the alpha-ketoglutarate and it turns it into a new metabolite called 2-hydroxyglutarate, or 2-HG, which shouldn't be there. It's a new metabolite. 2-HG builds up to huge levels in the cell and actually reprograms the cell and turns it into cancer. And so we call it an oncometabolite. So this was very interesting, but also it suggests a therapeutic approach, which is to try to create inhibitors of the mutant form of IDH to block the production of 2-HG and return the cell back to normal. And so that's what we've done. We have inhibitors of mutant IDH1 and inhibitors of mutant IDH2, and both are going to the clinic. So this was a really interesting cancer story, and that's what we've been working on for a while. But a couple of years ago, shortly after we published this, we learned that there's actually another condition where you get very high levels of 2-HG. But in that case, you get it all over your body and from birth. And that is a condition which is an IEM called D2-hydroxyglutaric aciduria, or D2-HGA. So this is a very rare and very severe disease. It's uniformly fatal in about the first decade of life. It's progressive and neurometabolic. There's been about 100 cases ever described. In half of those cases, about 50 cases, they didn't know where the 2-HG was coming from. So they had no way to describe the cause of that disease. So when our paper came out and described the function of this mutation, Cornelis Jacobs and his lab in Amsterdam saw that, and they said, aha, there might be a connection here. And they went to those patients, and they sequenced those cells, and sure enough, the 50 unknown patients have the exact same mutation in their entire bodies as the cancer patients did in their tumor. So this was a fascinating sort of story of two fields colliding. It also, of course, quickly suggests that there might be a therapeutic opportunity to use this drug that we were developing for cancer for these patients. And so that's exactly what we're pursuing. We're proud to publish this week at the ICIM a bunch of work in that area. So first, along with the Jacobs lab, we've shown that in cell samples from these patients, we can actually add the drug, and indeed it lowers the 2-HG levels in those cells by 98%, back down to normal levels. And so obviously that supports the idea that these drugs might have some benefit for these patients. In addition, we really want to study the disease more and understand it. And so you'll hear later about animal models. We agree that animal models are critical. And so we have a group led by Shengfeng Jin, who's here in the audience. You may have met her today, who can actually create animal models of diseases by taking a normal mouse and what they call knocking in or putting in the genetic mutation that causes a disease and seeing what happens to the mouse. And in this case, what happened is it reproduces the disease very nicely, and it gives us a great model system to better understand this disease going forward. We're not done with the science, though, because actually this is such a rare disease that there is no patient group, there aren't a network of investigators yet. This is really a true orphan disease, and so we're reaching out as much as we can to everyone in the world to say, have you seen these patients? Help understand the epidemiology. We're going to sponsor the very first natural history study and registry in this disease. and we're trying to reach out to physician advocates and patient advocates to the extent that we can find them. Because if the patients are out there, we want to help them, and we think we might be able to. So now let me tell you a second story about a different program that is even more similar to what we might be able to do in CDG. So this is pyruvate kinase deficiency. So this is a rare form of hemolytic anemia, okay, which is caused by mutations in an enzyme called PKR, pyruvate kinase R. So PKR is the last step in glycolysis, okay, which is a metabolic pathway that takes in sugar and turns it into energy and other things. In this case, I show energy as ATP, which is a molecule that carries energy. So your red blood cells have only one source of energy, and that's this pathway. Okay? Other parts of your body can get energy from other places. So patients who have a deficiency in PKR can't make as much energy as they would like. So that pathway, there's a big traffic jam up here, and there's not a lot of traffic going out here. And the red blood cell gets very weak and actually breaks apart very early, far earlier than it should, in a condition called hemolysis. It's the breaking up of red blood cells. So this is a very severe condition. Patients have lifelong anemia. They have to get a lot of transfusions. They get iron deposits. They have to have their spleens removed often. So it's a very severe condition. What is interesting, though, is we have an ability to reactivate this deficient enzyme using an allosteric activator that we've invented. And that's called AG348. So what this does, so as with CDG in the PMM2 example, this enzyme is never gone. You can't have 0% of PKR. You probably have 10 to 20% of the activity. And so what we can do is we have an enzyme that binds to a novel site on the protein and actually stabilizes it and boosts its activity. Okay, so it drives much more traffic through that pathway than it would do otherwise in its disease state. And so as a result, and I'll just show you what that looks like from the data, if you measure just the actual activity of the enzyme, as we add more of the drug, you can see that the enzyme gets more active. So that's doing what I said it would do, by about 7-fold, actually. And then you say, well, okay, did the traffic pattern restore? Well, we know that the thing we care about here is ATP, or energy. So we have the green line is what a normal patient would normally have. And so indeed here, you start low, but if you add more drug, then you start to reapproach the normal level. So this is all preclinical lab testing, although this was done in patient blood cells. But the next step is to try it in clinical trials. And so that's what we're doing. We should be in the clinic next year with this. And again, as with the other disease I mentioned, we're doing our best to reach out to clinical, scientific, patient communities. We are going to launch, again, the first natural history study. And we just pulled together the first patient summit in 2013. I'll preview what I'll say later, but I just want to say that, again, what this community has in CDG does not exist for many other IEMs. And it's a tremendous asset. I think we are going to be lucky if we can pull off in some of these other diseases what Vanessa and others have pulled off in CDG so far. So now let me finish by talking a little bit about our search for new IEMs. So this is a conceptual vision of a cell's metabolism, all the different pathways as they go around. And as we know, there are 600 diseases of mutations in these pathways. And so we're looking for diseases that have certain features. So one is severe, that have high impact, often pediatric impact. The second is neglected. As I said, we want diseases that don't have good therapeutics yet, where we can then put our time and energy. Third, they should be clinically feasible. That's an important one. So there needs to be a definable patient group, usually genetically. There needs to be enough epidemiology, or there needs to be enough patients out there that we can do trials. Because as was said before, doing really good trials is critical to showing that these drugs are going to work. And we need to be able to identify, are there biomarkers or clinical endpoints that we can use, much as Thorsten was saying, to say, do I see an early signal that my drug is on the pathway? And then can I follow clinical signs later to prove that it's having a good effect on the disease? So we want to be able to see what those might be. And finally, we care a lot about transformative potential. We want to have a hypothesis that a small molecule can make a big difference in both correcting the metabolic dysfunction of the pathway, getting the traffic flow going again, but also potentially have a disease-modifying impact either stabilizing or, if possible, reversing a large portion of symptoms in the disease. So how have we gotten to know the CDG community? So as I said, we're exploring many of these diseases, but CDG is one we're quite interested and passionate about. I think the last few speakers have done a pretty good job already of summarizing the fact that there's actually a bunch of ideas out there in CDG, especially in PMM2 so far, for using small molecules, and we're interested in all of them. Certainly we think allosteric activation of PMM2 is possible. We've talked about downstream supplementation of metabolites if we could create interesting prodrugs. There's also the possibility of blocking certain parts of the pathway to drive more flux overall through what is functional in the mannose pathway. And I think all of those are good ideas, and if there are more good ideas, we'd love to hear them. I think one really important point that this field has that's key is that experience with mannose in MPI, as well as some of the more recent emerging experience. Because what those show is that it didn't take much. A little bit of extra flux through that pathway in that disease had a profound impact on those patients and really did transform that disease. So I think with something like PMM2 CDG, we can't do that simply with sugar, but we're hoping to find some sort of novel therapeutic approach to get just a little bit more traffic going through that pathway in a way that we can hopefully have the same kind of profound effect for those patients as well. I've already mentioned that in this field so far we're collaborating with Dr. Freese's lab at the Sanford Burnham, but hoping to get to know a lot more people as well. And again, the goal here is to explore as many of these approaches as possible and see whether we can help in this disease. At the very least, I think it's exciting that we're here and Mark is here and others are doing interesting work, because I think the more people come to the table with good ideas, the more progress hopefully will be made. So before I finish, I just wanted to give a few thoughts to some of the conversations that came up yesterday about what can we do, what is the role of the community here, and how do we get to therapeutics, because it will be a long road to get to a novel therapeutic like this, and we have to be aware of that. So just a couple of thoughts. One is it does begin with science. I think we need to have a fundamental understanding of the basic function of the pathway in order to build on that into a therapeutic. And so I think that basic research, the story that Hud was saying, is absolutely critical. But then, in a rare disease, leadership and advocacy from patients and families is absolutely critical to making the rest of it happen. Because nothing will come together without that. And I think you've heard that over and over again, but I just want to reiterate it one more time. They don't quite come together yet. We're doing our science. You guys are building the momentum. But I think it doesn't take long to start bringing them together. And then you get to the third part, which is a big partnership. I think that at the end of the day, developing a new therapeutic will be the marriage of patients and families, physicians, researchers, industry. At some point, the regulatory agencies and other types of groups will get involved. And the more we build a community around these types of transformative therapies, the better. And so I think the work that has been going on here at the patient and family level is just absolutely foundational to that. And the more that it continues to build momentum and go global, I think the better, because there is power in numbers, and it'll continue to draw attention and momentum to the space. So with that, back to wrapping up for Agios. So I think we have a phrase internally. We try to hire great people, think big, have fun, follow the science, and do what's right for patients. And we actually use those last 2 points as decision-making rules. So if we have a decision to make, we basically come down to what does the science tell us to do and what's right for patients. And if we do those 2 things, we're pretty certain that good things will happen. So with that said, let me wrap up. I just want to, again, thank you all for having us and making us feel so welcome. It's been an incredible experience. Our whole team is here. I think we've learned a ton, and we're more motivated than ever to keep looking at this disease and see if we can help in any way. So thank you very much.